Well, it's wonderful to be here today, and I really am so grateful for the opportunity and the invitation to be able to start the day off for you all. And hopefully I'm able to share with you a story, a journey that you can resonate with. And I suspect most of you in this room have a child. If you don't, you're touched by a child or you work with a child. Neurotypical maybe. But I want you to just take a moment and think about that child or children. And I'd like you to ask yourself, what wouldn't you do for your child? I'm going to walk you through a journey of drug development, focusing on cell and gene therapy and gene editing. And I'm going to use one example of a monogenic autism spectrum disorder called Angelman syndrome. I'm the Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics. And as Allyson said, I co-direct a biomarker and outcome measure consortium in order to understand measures in clinical trials that are both meaningful and measurable to patients and families. I'm the Chief Development Officer at an accelerator called AS2 Bio, and I'm a strategic advisor to almost every neurodevelopmental disorder of monogenic conditions that are working toward developing therapies for these monogenic conditions. I started off as a veterinarian, and I still function as one. I'm an interventional radiologist and an internist, and I've been working in medical device development for my entire career. Until 2015, when I became the mother to this beautiful little girl, and her name is Quincy, and she lives with Angelman syndrome. So I'm going to walk you through a journey. That journey is a patient journey, a clinician journey, a community journey, an industry journey, and the holistic journey of what it takes to make a difference, because you can't do any one of these things in a silo. I received a phone call in 2015 from a neurologist that said, "I have catastrophic news for you." The next thing I heard was Angelman syndrome. And I asked, "How do you spell that?" I have no idea what this man just said. And I did what every parent should never do. I did what any clinician and doctor would advise their patients to never do. I Googled it. And I opened Wikipedia, the second thing I shouldn't have done. And I read all the things that Quincy would never be able to do. She would never walk, she would never talk. She would not sleep more than 2 to 3 hours a night. She would never live an independent life. She would not have friends. She wouldn't go to school. She certainly wouldn't go to college, get married, and have children. Your 5-month-old baby girl, her life was just destroyed. In the words, "I have catastrophic news for you," because she had some developmental delays at 5 months of age. And my world changed. Then I thought, well, what will she be able to do? What is she going to be capable of? And I learned that it is not degenerative. Her prognosis for survival is excellent, but her quality of life will likely be nothing that I dreamed about for my little baby. How do I give this child the best life that she's capable of? And I realized very, very quickly, just like Allyson, I had to advocate for Quincy. I had to learn to execute if I wanted to change the trajectory of what I was reading on Google, because it was not ready for any child to have a treatment. And then I went through a moment within 30 seconds of reading this, falling to the floor. I can't do this. And I don't really want to. I didn't sign up for this. Like most of you, I'm a type A personality. I went to vet school 2 years early. I wanted to be a vet since I was 8. I was finally a professor doing exactly what I wanted to do. I wrote my first textbook. I published my 100th article. I was training interns and residents at the largest animal hospital in the world. I was exactly where I wanted to be in my 30s. And I had an entire career ahead of me that I was never ready to give up. I planned everything in my life exactly the way I wanted it to be, including having a daughter. And I had one. I actually had 2. And I fell to the floor in desperation that how is this now my life? And then I went through this moment within another 30 seconds of what kind of mother am I? I've been given this baby girl. It is my responsibility to care for her, and I don't want to. How is that fair to her? It's not her fault. It is a random genetic disorder. How do we handle this as a family? She had an older sister. How do I navigate this as a wife? I have a husband, and I was now pregnant with her little sister. I needed to figure this out, and I needed to figure out how to manage a world of a disability that I've never been touched by. I had no idea where to go. I had no idea what to do. And I did realize there has to be a solution to this problem. Because as doctors, we all know knowledge is power. And things are either possible or impossible, and there's no in between. And unless I can prove this is impossible to fix, I have to try and fix this. I needed to be brave, and I needed to be smart. And I became a clinician very quickly. I called every senior author I could on every publication. I attended every scientific conference I could. And something was so incredibly clear to me with the incredible science that had been funded by the foundations and the NIH in Angelman syndrome research. So much work had been done. A gene therapy had been tested in a mouse model showing that there was rescue of the behavior of the mouse. I was so excited to realize it was a really great decade to be a mouse with Angelman syndrome. But it definitely was not the right decade to be Quincy with Angelman syndrome. Translational science is outside of the wheelhouse of most scientists. And early science and discovery and proof of concept is not something in rare monogenic disorders at this time that pharmaceutical companies were interested in. The clinicians wanted to help my daughter, but they didn't know how to develop a drug. And the scientists were really interested in incredibly important biological questions, but didn't know how to develop a drug. So this is called the valley of death, where you're sitting in between. There's the potential, but there's not the reality. And this is the world that I lived as a veterinarian. And if I could do one thing, I could help develop and advance a treatment for Angelman syndrome, because as a veterinarian, that's exactly what I did. I developed medical devices for animals with naturally occurring diseases where there was a clinical unmet need. And those devices are then translated to use in the pediatric population where medical devices are underserved in kids. And that's what I had been doing for the prior decade. So I learned everything I could about Angelman syndrome, and I'm not here to give you a genetic lesson, but I think it's important to understand what I understood at the time, because that's how the plan happened. So we all have 23 pairs of chromosomes, 46 chromosomes. Quincy has a single defect on one allele, the mother's copy of chromosome 15. And if you look at this in a schematic, what's really interesting about this, and this is going to help you understand what we did and why, is that if you actually look at the schematic, in all of us, in me, in you, and in Quincy, the paternal or the father's copy, we have one copy from mom and one copy from dad. The father's copy in all of us, only in our neurons of the brain cells, are silent in that this one single gene called UBE3A is silenced by this long non-coding RNA called the UBE3A antisense transcript. And the maternal copy is read in all of us. So we are only living off of one copy on the mother's side that's being read. But in Quincy, the mother's side is missing. So she has a healthy copy on her father's side that's silenced like the rest of us, but she has a missing copy on her mother's side. And therefore, this protein is not being made at all in the neurons of her brain because she doesn't have that single copy that's reading. And therefore, it seems logical to the rest of us that we would be able to say, maybe we can replace that gene. So the symptoms of Angelman syndrome are dramatic. Complete lack of speech, not a single word, sleeping 2 to 3 hours a night, having debilitating seizures, and never living an independent life, just to name a few. But the impact that that has on a family is dramatic. Stress, fatigue, loss of work and employment, the unbelievable financial impact on a family, it is tremendous. And we can't take that for granted. But something that happened that I realized right after her diagnosis was that there's a population of kids with Angelman that have mosaicism, meaning they have some cells that read maternal UBE3A and some cells that don't. Those that have only 1 to 5% of UBE3A in those cells have speech. They walk, and they have very few seizures. Kids that have 20% are speaking in sentences, and kids with 40% are considered neurotypical. What does this mean? If we can give my daughter 1 to 5%, that's a different child than I live with today. Imagine that. And so we didn't need a lot to go a long way. So I joined the board of directors of the foundation. I became their CSO. And I really helped to lead a plan and a path. We needed a roadmap. How are we going to get there from mouse to human? We created a plan and a path that cost $5.8 million. We raised that in about 6 weeks. And we had a commitment that we were going to do this in 24 months, which seemed very ambitious, to say the least. But guess what? We did it. We came up with many candidates, human candidates, one of which within 6 months, and I'll share that with you. One option is you can replace the gene or replace the protein. The other is you can unsilence the silent copy of that gene or protein. Or you can think about the effect downstream that that protein has on the symptoms of these children. And you need to also be prepared for running these clinical trials. And so this is what a gene therapy looks like. This is an Angelman mouse that looks a bit like my daughter on how it walks and how it's incoordinated. And this is after gene therapy. A single mouse, you can see the dramatic difference. And this is a hematopoietic stem cell gene therapy in an adult mouse. So this mouse was treated as an adult and had full rescue of its symptoms. This is a gene editor. By editing and unsilencing that copy, you can see the Angelman mouse in the middle, the wild-type mouse, the typical mouse on the left, and the treated Angelman mouse on the right. The treated Angelman mouse on the right is doing better than the wild-type mouse. It's Superman. And that's what gene editing could do. In a single mouse, you can see the difference. It's that profound how therapeutics can impact these kids. And so you don't need to look at this. All I want you to do is look at this heat map, that various different therapies can have various different impacts. And this is the antisense oligonucleotide that I just want you to look at very quickly. So as we prepare for clinical trials, we have to think about what's the trajectory? What's the trajectory of this disorder, and what do we see over time? Over 18 years, you can see how flat these kids are. This is very important. We have an 18-year natural history study, and we have to try to beat that trajectory. So we develop endpoints and biomarkers over the last 10 years that have really transformed how we're going to run these clinical trials. And this is just an example of the amount of work that went into that. And in 2017, everything changed. Within 6 months after that $5.8 million grant, we had an ASO, an antisense oligonucleotide, a human candidate, and we decided to start a company. We decided to start a company called Therapeutics, and we were going to take this forward into human clinical trials in a hurry to get this to human patients to see proof of concept in humans. And that's exactly what we did. So I started this industry journey, which took us on a ride, a wild ride, and we got this approved through the FDA in 13 months. By 17 months, we were dosing our first patient, and we have data to support it. The program was so successful in its early stages in an open label study, so we'll see what the placebo looks like. Ultimately, it got acquired by a company called Ultragenyx, which allowed an infusion of funds into our foundation, since the foundation owned a majority of this company. This is just some early data in that trial, and all I want you to see is the flat of the natural history study in communication, the flat of the natural history study in cognition on the right, and on the left you could see the slope of that trajectory just going wild as these kids are gaining skills right before your very eyes. These kids that had not a single word speaking, kids that could not swim swimming, kids that could not self-feed feeding, kids that couldn't sleep more than 3 hours a night sleeping through the entire night for 12 hours. And so that's the ASO. Look at that heat map. There's a lot of red and very little green. Look at the gene therapies and the gene editing and how much green there is. We only expect more and more as we get these gene editing therapies into the clinic. And so ultimately, drug development is hard. For every 15,000 drugs, one gets approved. For every period of this roadmap, it costs up to $250 million for one drug to be approved. Our foundation funds this $15 to $25 million space in order to make sure that nothing dies in the valley of death and that we can have human proof of concept to move these things forward. So when Quincy was diagnosed, there was 5 academic programs, which went to 15 after that $5.8 million infusion, then to 34, and then the stock market crashed in 2022. 34 went to 24, 24 is now sitting at 28, fully funded by our foundation. So 5 programs in clinical trials, 4 in IND-enabling studies, and our first gene therapy will be in the clinic this year. So it's really important to realize we have to accelerate all shots on goal, and traditional drug development has failed rare disease communities. but it's not failing this one. And because of that, we're ending with an accelerator that we are advancing all of these programs ourselves. We're not relying on government money because, as you can see today, that may not be there for long. We cannot rely on industry because they may not be there for long. We have to rely on ourselves, and that's exactly what we're doing. So this is where patient advocacy goes to venture philanthropy, to become disruptive, to how do you develop drugs for these therapies. From bench to bedside, we've invested over $60 million in this research to create a matrix of how we can be the low-hanging fruit and be successful. So this is just one example of how we can do that. And these are all of the programs we're doing by ourselves, without the support of the traditional drug development ecosystem. And so this is just one example in one disease of how to make the impossible possible. And I hope this serves as a huge example for the thousands of other monogenic disorders, hundreds of other autism spectrum disorders that are similar to Angelman syndrome, that can see similar success and hopefully a heck of a lot more. So thank you for listening. This is just my reason, and this is what I do for my daughter.